Great. Next slide, please. Yeah, we're one of the original gangsters. We began in 2004, so we've been operating for now over 20 years. And in terms of what changes have occurred in primary ciliary dyskinesia, I can say very briefly everything. We had just the basic understanding of the disease and the pathophysiology, and through the efforts of the consortium and our international colleagues, we have been able to define the cardinal features of primary ciliary dyskinesia, so we're no longer guessing who has the disease. We've been able to characterize the earliest origins of lung disease. Probably the place where we had the greatest impact is out in developing better diagnostic tools. We are no longer reliant on transmission electron microscopy to make the diagnosis. We still do it, but it's not a primary assessment tool now. So we've established that nasonitric oxide concentrations is a very useful screening tool for people with primary ciliary dyskinesia. We defined the prevalence of respiratory pathogens and pulmonary function decline primarily in childhood. Because this involves the upper airway as well, we have characterized the otolaryngologic manifestations of this disease. One of the more interesting aspects of this is when we first started, there were only 2 genes that were associated with primary ciliary dyskinesia. And currently, the ClinVar gene curation expert panel is now assessing nearly 70 different genes that are linked with this disease. And in the process, we've discovered that one, not everyone with primary ciliary dyskinesia is the same. And second, there are genotype-phenotype relationships in this disease that has provided some remarkable insights into the pathogenesis. We've also, in the process of trying to determine the genetics of primary ciliary dyskinesia, we have found genes that are uninvolved with cilia, specifically WFDC2, that causes clinical features that look for all intents and purposes like primary ciliary dyskinesia, but it's not. We've also identified overlapping immunodeficiencies with PCD, and in the process, we've developed both North American, but also international diagnostic and treatment guidelines. In the process of getting smarter about this disease, we have developed and validated some quality of life surveys that are being used or have been used in clinical trials in primary ciliary dyskinesia. And in fact, there have been 2 clinical trials that I can say with confidence was springboarded, if you will, because of the genetic disorders of mucociliary clearance consortium, including an ongoing study looking at liposome encapsulated mRNA therapy to treat people with DNAi1 defects with primary ciliary dyskinesia. This is being run at 3 sites here in Chapel Hill, as well as in Freiburg, Germany, and Copenhagen, Denmark. Next slide, please. And so, one of the things we were asked when we put in the original, when we put in the proposal 5 years ago, was how are we going to sustain this effort? And this largely is because of our relationship with our patient advocacy group partners, particularly the PCD Foundation. In the process of working with the PCD Foundation and what we've learned through the consortium, we now have 60 PCD Foundation clinical centers across North America who know what they're doing when it comes to this rare disease. We've developed and maintained a PCD Foundation patient registry to help us identify patients for future clinical trials. And we are also in the process of creating the North American Primary Ciliary Dyskinesia Network for Clinical Trials, which is going to build on the consortium to facilitate efficient, effective clinical trials, observational, translational research programs, patient engagement, and quality improvement initiatives. Dr. Berry, who just spoke, about 20 minutes ago, is absolutely spot on. One of the interesting things that has occurred, and I have to give credit to the consortium and credit to the funding of the consortium, is that suddenly industry has discovered primary ciliary dyskinesia. And so now we're being approached much like we saw in cystic fibrosis 20 years ago, we're now being approached by pharma to do clinical trials. But it's a rare disease. And so it's going to be really important for this network to not merely coordinate our efforts in North America, but also coordinate those with our colleagues in Europe. So we have enough patients to ensure that these trials can be completed. And I'm going to stop there. I wish I had a picture slide showing everyone who participated in this. I apologize. There you go.